This is the chapter overview of the blood, chapter 17. General overview. In males, there's about 5 to 6 liters of blood found, and then in females, there's about 4 to 5 liters. Physical characteristics include the pH of 7.35 to 7.45. This is a very important number because if the blood pH drops anywhere below this or goes anywhere above it, you know that there's something going on in your body. And then the temperature is about 100.4 degrees Fahrenheit. The major components of blood, there's 2 major components, which include formed elements or cells, and then the plasma. Under formed elements, there's erythrocytes, which are also red blood cells. This is the most abundant. There's leukocytes, which are white blood cells, and then there's platelets. The second major component is plasma, which is basically the fluid matrix. It's 55% of the blood volume. This includes stuff like solutions of salts, glucose, amino acids, vitamins, and stuff like that. Functions of blood. Blood distributes oxygen and hormones throughout the body and also removes carbon dioxide. It also helps to regulate temperature, pH, and it maintains blood volume. And then the third thing, it protects because blood clots in order to prevent against blood loss, and they also use antibodies to help against infections. Blood plasma. Blood plasma is composed of 90% water and 10% solutes. The solutes in blood include proteins like albumin, globulin, clotting agents, hormones, and enzymes. They also include nutrients, which is glucose, amino acids, fatty acids, and glycerol. Thirdly, they include electrolytes, which is charged ions, and respiratory gases, which include small amounts of oxygen and large amounts of carbon dioxide. And then lastly, they include non-protein nitrogenous substances like lactic acid, formed elements. So now we're going to be talking about red blood cells, or erythrocytes. The structure of a erythrocyte is a biconcave disc that does not have a nucleus. This helps because they can fold up as if they're a taco, and they can easily go through the small vessels of the body without getting stuck. Spectrin is the protein that forms this biconcave disc shape. Red blood cells usually last up to 120 days, where they soon are recycled after that. So red blood cell production, or erythropoiesis. It starts with hematopoiesis, which is the blood cell formation. It occurs in red bone marrow. The most important part is to realize that whether it's red blood cell or white blood cell, they're both formed by a hemocytoblast at the very beginning. This forms all formed elements, which includes the red blood cells and white blood cells once again. Regulation of erythropoiesis is based on hormonal control with erythropoietin, and then the dietary requirements with iron, vitamin B12, and folic acids. Without these requirements, you'll see clinical problems that we will go over in the next slide. And then, like I said before, the lifespan of red blood cells is approximately 120 days until they get old and rigid and are recycled into the spleen. Clinical problems of red blood cells. Anemia is the reduced oxygen carrying capacity. So you'll see in the picture below that there's a normal amount of red blood cells and then the anemic amount of red blood cells. So you can see there's a very big decrease in the amount of red blood cells in anemia. This can be caused by 3 different things: the decreased number of red blood cells, decreased hemoglobin levels, or abnormal hemoglobin levels. And then there's also polycythemias, which is a high level of red blood cells. Formed elements, so white blood cells we're talking about now. So leukocytes or white blood cells, you'll want to know both names once again. They are the complete cells with a nucleus and organelles. The main function is immune defense. The types of white blood cells include 2 different types and 5 different kinds. So there's granulocytes, which contain granules. These are neutrophils, which are the most abundant in the body, eosinophils, and basophils. And then there's agranulocytes, which lack the granules. These are lymphocytes and monocytes. So now on the next slide, we're going to go over what they look like and what the functions of them are. So starting with the agranulocytes, you'll see the monocyte up at the top. This one usually has a U-shaped nucleus, and they phagocytize at chronic infections. The next one is a lymphocyte. You'll see a little sliver of the cytoplasm sticking out towards the right. These guys fight infections and make antibodies. And now for the granulocytes. There's the basophil, which has very purple pigmented all over the nucleus. It's very purple under the microscope. These guys produce histamines. And then there's eosinophils, which is normally bilobed, and unlike this picture, normally under the microscope, they're very red pigmented. These guys kill parasitic worms and have antihistamines, produce antihistamines, which makes them antagonists to the basophils. And neutrophils are multi-lobed. They phagocytize bacteria at acute infections. Clinical problems of white blood cells. There's leukemia, which means there's too many white blood cells that are misshapen and non-functional. And then there's infectious mononucleosis, which we know as the kissing disease back in the school days. And then there's leukopenia, which is the low-level white blood cells, which is the opposite of leukemia. There's hemostasis, which keeps everything intact. This occurs with vascular spasms, which is vasoconstriction at the site of injury. But it also happens with platelet plug formation. This is a very easy process. It's at the site of damage, blood vessel endothelium. The platelets swell, they get spiky and sticky. I like to think of them as like little sand spurs. So the platelets stick to exposed collagen at site of damage, and then they degranulate. The chemicals released during this process is serotonin, which increases the vascular spasm, ADP, which attracts more platelets and stem them to degranulate, and then prostacyclin controls the size. So it's important, or he doesn't need us to know all the steps of the intrinsic and extrinsic pathways, but it's important to realize that both these pathways get to the same conclusion. They both form prothrombin activator. The intrinsic is a little longer process, and the extrinsic is quicker. So here's an overview of the intrinsic pathway. And again, this is the extrinsic pathway. It does form the prothrombin activator at the end, and you'll see that it is a lot shorter than the intrinsic. So once a blood clot is done, you need the clot retraction and repair. Actomyosin protein complex and platelet-derived growth factor helps in this process. Fibrinolysis removes clot whenever it's no longer needed. Plasminogen, tissue plasminogen activator, and plasmin aid in this. Control of clot formation is localized and very strong. Heparin and antithrombin 3 aid in this one. So blood transfusion. This is where our blood types are needed, because with a donor and a receiver, there needs to be the same blood type, or else a transfusion reaction will occur. And there's a blood typing game online that will help you in your blood typing. Clinical problems. Hemophilia lacks certain clotting factors. This usually comes from mom because it's an X-linked factor. Then there's thrombocytopenia, which is a low platelet count, liver dysfunction, not making procoagulants, and thromboembolism, which is clot blocks off the blood vessel. And that is it for the blood chapter. Thank you.